Hi. So I'm going to be talking about the microstructure of the cortex itself and how we can link post-mortem measurements to in vivo measurements of cortical structure. So the issue is that with in vivo MRI, we can measure large numbers of individuals and really reliably generate biomarkers, but the resolution is relatively low and the source of the signal is quite ambiguous. So if you're interested in regional differences in cortical thickness, it's not clear whether these are driven by changes in myelination of the cortex, or if it is a histological change in cortical thickness, which layers might be driving this. At the other end of the scale, with histology, we can do very high resolution. We can stain very specifically for particular neurons, and you can absolutely see the layers. However, there are often limitations in that they're done in 2D, with small numbers of sample sizes, often requiring manual annotations, which causes its own set of problems. So in the talk, I'm going to be talking about how we've been automatically generating segmentations of the histological cortical layers in 3D, first through developing a profile-based approach, then a package to generate volumetric surfaces on which we have a poster at OHBM, and then finally deep learning segmentation of the 6 cortical layers across the whole cortex to generate an atlas of the histological layers. And then I'll be talking a little bit about how we've used this atlas to explore patterns of cortical thickness that we first identified in MRI, but now looking at which layers might be driving these changes. So to do this work, we've been working on the big brain, which is a 3D histological model of the human brain. It was a 54-year-old man who died. The brain was fixed. It was then sectioned, you can see in part D, to 20 micron thick sections. Each section was digitized and then reconstructed into 3D. So we have a histological model where the voxels are 20 by 20 by 20 microns. And you can see that this is sufficient resolution to start picking out the layers of the cortex. The first step was to generate white and gray surfaces like you have with FreeSurfer or Civet, where each point on the white matter surface has a corresponding point on the gray matter surface. This enables us to generate intensity profiles for every vertex across the surface by sampling 100 points between the peel and white matter surfaces. And so here on the top is one example of an intensity profile extracted through the cortex, and below is the first derivative of that same profile. And what you're doing is looking for features within these intensity profiles that consistently correspond to cortical layers. And the first most obvious feature that we found is if you look at the first derivative, this big peak, which corresponds to the boundary between layers 1 and 2. And so you can take each profile, identify that peak, and you end up with a surface that follows that boundary between layers 1 and 2. And the second feature that we looked for is this dark band you can see in the histology, which corresponds to this broad peak in the middle of the profiles. Again, you can identify that peak on all of the profiles, and you end up with a surface that follows somewhere around the bottom of layer 3, middle of layer 4. Now you have an algorithm that you can run on every single one of the profiles across the entire cortex, and you end up with a 3D model of the cortical layers for both hemispheres comprehensively. And if you look a little bit at this model, it's quite small, but one of the features you can see is that this layer 4 purple surface is much more superficial in the gyrus and much deeper to the white matter in the sulcus. But because we have 3D mesh reconstructions of these, we can start quantifying that effect. So we can compare the depth of layer 4 to the curvature of the surface. So in the top left, that's just depth of layer 4 against mean curvature, which describes whether you're in a gyrus or a sulcus. And if we overlay that on the cortical surfaces, you can see that the sulci, layer 4 is much deeper, the red color, and on the gyri, it's much more superficial, green or blue. However, from the histology, there's a model for how the curvature might affect layer depth, which is called the equivolumetric model, which assumes that a layer takes up an equal volume in an adjacent gyrus and a sulcus. And if you use this model to predict the depth of layer 4, you can see that you very accurately account for local morphology, and all you're left with is the regional differences in the position of layer 4. And this is the first important point for in vivo imaging. If you're trying to sample in fMRI or structural MRI the intensity of a particular layer within the cortex, if you just take a mid-thickness layer, you will be sampling a different layer in the gyrus to what you are sampling in the adjacent sulcus. Whereas if you take an equivolumetric approach, you'll be sampling the same layer in the two pieces of cortex. And this is a poster we have now at OHBM. Based on your peel and white matter surfaces generated either through physurfer or civet, you can generate these red equivolumetric surfaces. So this is on the big brain histology, but it works the same on MRI surfaces as well. So this is poster 689 on Thursday. So we have 2 layers now, but what we really want to do is generate a 6-leg segmentation of the whole brain. And to do this, we went to deep learning because the cytoarchitecture of different regions varies, and that means that the profile features that correspond to cortical layers vary. And deep learning is very well suited to learning the features that correspond to particular cortical layers. So to do this, with the histologists at Jülich, we segmented 60 training areas. This is an example of 2. The different grayscales correspond to the different layers, 1 to 6. And so they were taken from front to back, representing a variety of cortical cytoarchitectures. This was registered to the big brain, and then we can generate a large amount of training data. So from each of these sections, you can generate tens or hundreds of thousands of training profiles where you know the raw intensities of the profile, and you have a smooth version to give some context, and you have the ground truth segmentation as done by manual segmentations. And so with 500,000 training profiles, we trained a neural network to recognize layers from anywhere in the cortex. That means we can now test it on the full profile data set, which generates a full 3D segmentation of the 6 layers across the cortex. This is the original coronal section, and the key thing that we can now do with the big brain is that pieces of cortex that were previously very obliquely sectioned that you couldn't possibly have segmented, we can now segment because we can extract profiles in any oblique direction. Just going to zoom in on one of my favorite parts, which is here in the occipital cortex. This is the colored lines are the automatically generated layers, and the gray lines are the manual segmentations, and this is two more segmented areas. And if we zoom in here, this is the boundary between V1 and V2. And V1 is very unique in having a very thick layer 4. And you can see this in the manual segmentation. This band here goes from very thick to very thin. And in the automatically generated layers, you also see this very abrupt change. Green and blue suddenly get very far apart in V1, and this continues outside of the training region, and V2 with a much thinner layer 4 is present here. So based on the profile alone, the network is able to recognize that this is V1, and it should have a thick layer 4. Now that we have these 3D surfaces, we can start looking at patterns of histological cortical thickness. As a first validation, we wanted to check these against the von Economo maps. So this is von Economo's measures of total thickness, color-coded. Darker is thicker, and white is much thinner. And we see the same pattern in the big brain. So one of the crucial features is that the motor cortex here is one of the thickest areas, in both sets of brains. And the occipital cortex is very thin in both von Economo and our data. Another feature that's really nice about the big brain is that von Economo is at a relatively coarse scale. You don't see a lot of local variability, whereas you can see the differences between gyri and sulci on the big brain because of the high density of measurement that we have. However, now we can go a step further and say which layers are driving these changes in cortical thickness. And so we can generate these maps for each of the cortical layers across the entire cortex on both hemispheres. Where available, I've included the von Economo map. It's probably quite hard to draw visual comparisons, but you can start to see patterns like layer 3 is quite thick in the frontal cortex. And here on layer 4, we don't have the map for von Economo, but what I've superimposed here is the segmentation of V1, the histological segmentation of V1. And you can see that if you almost just threshold the layer 4 thickness map, it would give you this segmentation. So that's how accurately that boundary is being defined by the network internally. And now I'm just going to try and link what we have from the MRI to this histology. So looking on MRI patterns of cortical thickness, we showed that as you move away from the primary sensory areas, the thickness of the cortex increases. So This is geodesic distance from V1, and this is cortical thickness. This is the group statistics, and these are the individuals. So every individual shows this pattern of increasing thickness as you move away from the primary visual, somatosensory, and auditory cortices. However, we didn't know whether this was driven by a myelin gradient, or if it is a thickness gradient, which layers are driving it. Now, with the big brain, we can answer this question. So on this plot are the gradients. On the top is the whole cortical thickness gradient, and so you can see that the visual, somatosensory, and auditory, orange, red, and green, all exhibit a positive thickness gradient in total thickness, but this thickness gradient is driven not uniformly across the cortex, but primarily by layers 3, 5, and 6, which happen to be the pyramidal neuron layers. By contrast, if you look at the motor cortex, so if you're going anteriorly from the motor cortex, you see the inverse gradient, which is what we'd expect. The motor cortex is thick, and it gets thinner as you progress anteriorly. So there's an inverse pattern between what you see with the sensory gradients and what you see with the frontomotor cortex. What's really nice is that now we can link this pattern from the mesoscale of the layers to the microscale of particular neurons, because it's the pyramidal neuron layers that are driving this, and individual specific cell morphometry studies have shown that as you move across the visual cortex, the arborization of the pyramidal neurons increases. This would cause an increase in the volume, the local volume of the cortex, and therefore the local volume of the layer. So we have now a way of linking macroscale MRI cortical thickness gradients to mesoscale layer gradients, all the way down to single cell morphometry with a causative chain of links. So that's basically what we can use the big brain for, to link the macro, the meso, and the micro scales of cortical anatomy. This 3D layer segmentation of the 6 cortical layers is now freely available to download at the big brain site. And the layers that we have now confirmed histologically can be used as prior models for MRI modeling of cortical layers. I'd just like to thank all my collaborators, University of Cambridge, at ULISH, at the Montreal Institute for Learning Algorithms, and the group of Alan Evans at the Montreal Neurological Institute. And thank you all for your attention.